So in this video, I'm going to talk about the things that control infectious disease, like what determines the likelihood a disease is going to happen, what determines how long it will last, what determines how severe it will be. Again, a lot of this stuff will seem pretty clear or straightforward. We just want to cover all of it, make sure you don't miss it, make sure you don't gloss over something that might actually be different from what you think it is. It's hard going through these things because so many of them will be familiar. The hardest thing for a student is to identify a misconception and correct it, and that is a critical thing that students need to do in any course. It's identify when are you using a word in a different way than what your teacher wants you to use it as. There's no way to avoid coming to a class with misconceptions. That's just humanity. That's how it works. We learn things in the world and then we take classes. So look for situations where you have to unlearn something. Okay, enough of that. The determinants of infectious disease are how contact happens. So is there a meaningful exposure to a pathogen? How virulent is the pathogen? So how able is it to cause disease? And then how resistant is the host? Can they fight it off or can't they? Another way of thinking about this is the likelihood or severity or duration of an infectious disease is controlled by conditions of contact times the virulence, divided by the resistance of the host. So this is not an equation we care about. There's not a number we're going to be calculating, but this is the kind of idea. So if we think about conditions of contact, we're thinking about things like infective dose. We're thinking about how long the contact occurred over, and then how exactly did the microorganism get into the body. So if we start by infective dose, I've talked about this a little bit before, and this is the number of virions or bacterial cells necessary to cause the infection or the disease. So if you're exposed to a smaller number than the infective dose, you don't get the disease. If you're exposed to a higher number, you would get the disease. So why is this even a thing? Why isn't just one cell enough to cause the disease? There are a lot of answers to that. The pathogen might really be able to evade the host response. The pathogen might not be. So for example, a pathogen of the intestines has to be able to survive the acid in the stomach. And something like Vibrio cholerae causes cholera is not very good at that. So the vast majority of those cells that go through the stomach just die there and don't cause disease. But if you ingest a vast number of those cells, just through probability, some of them will survive and go on to cause disease. It could be that even one, like think about something like Staphylococcus trying to infect your dermis. It might be that one little spot of dermis that's infected with one cell isn't enough to cause disease, but maybe if there are 10 different sites near each other, that is. We won't think about most of these in detail, but just understand that it is a complex idea, and it is not predictable. That's the thing. In many situations, we wish we could predict these things, but we can't. So, as I brought up before, infective dose is highly varied. This is a figure that shows the U.S. Food and Drug Administration estimates of infectious dose for various pathogens and Health Canada's same estimates. And you'll see they're not identical. So in some cases, like the FDA says that cholera can be caused by 10 to the 6th, a little over 10 to the 6th cells. Health Canada, and that's like a minimum, and so that would be the most worrying number. Health Canada is publishing the whole range because there are different strains of Vibrio cholerae, and some are more likely to cause infection than others. We could contrast that with the nasty E. coli strains in Shigella, which are down around 10 cells or 100 cells. So these are closely related bacteria. Vibrios and E. coli are not so different from each other, but they do different things in the guts, and they have different abilities, and they cause very different diseases. So there's that. This is how a person would be exposed to these. Vegetables, anything we eat raw could expose us to these. Yersinia, which causes the bubonic and pneumonic plagues, is given to us by a bite of a flea. Streptococcus pneumoniae can be growing in your pharynx without you ever knowing about it, or you can inhale it from another patient who has it, and then you get cholera from contaminated drinking water. Virulence is a description of the pathogen. So it tells you how likely they are to cause a severe disease or cause a disease. We can measure this explicitly in the LD50. So what number of organisms kills 50% of test animals? And so a low LD50 would be like 10 cells. 10 cells kills half the mice you expose them to. Well, that's very low. A high LD50 would be like 1,000,000 cells or 1,000,000,000 cells. So a low LD50 is more virulent. Something that has a low LD50 is more virulent. We also have ID50, or the infectious dose of 50%, and that's measuring explicitly how many cells are needed to infect half of the test animals. And so this is more precise than the infective dose. The infective dose is a minimum. It's meant to be a minimum, whereas the ID50 is a repeatable, experimentally determined number. Whereas a minimum is harder to pin down. So it's hard to find infective doses. And that's all there is that. Virulence factors are traits that certain pathogens have, and when we're looking at a pathogen, we can list its virulence factors, and those are the things it has that make it more likely to cause a severe disease. So things like capsules, polysaccharide capsules on bacterial cells, allow them to evade the immune system. Bacterial endospores allow bacteria to pass through the gut or to survive in the environment. There are a lot of different enzymes bacteria will make that they use to directly attack immune cells or to release toxins and things. So toxins would directly attack the host and cause dysfunctions in the host that could be exploited by bacteria. And then the faster organisms grow, the faster they can reach overwhelming numbers that can defeat the immune response. We can think of several different mechanisms hosts would use to get around the host defenses. Protists, for example, can, as a normal part of their life cycle, will be cysts. And the ones that are encysted, these can go through the stomach without being killed by the acid, and then they can change into a vegetative form in the gut. Bacterial endospores aren't really for the same purpose. Usually, bacterial endospores are made when the bacterium is starving, whereas cysts are a normal part of a protist life cycle. But a bacterial species might not make endospores if it's never in a starving condition. Antigenic variation is a really nasty thing some pathogens can do to evade host defenses. So what's shown here are trypanosomes. This is showing trypanosomes that can change surface molecules. And so we mount an immune response to this one with its surface proteins and surface sugars. We can develop antibodies against it, and then its offspring can switch and change to different surface molecules that our immune system will not detect. And so this sleeping sickness will go through cycles of fever and chills each time a new variant shows up with different antigens on its surface. And Plasmodium, which causes malaria, is also an expert at this, and that also causes a cycle of fever. A lot of microorganisms attack the host immune system. So HIV, for example, destroys T cells. It's safe to assume that any virus that makes healthy people sick, so anything from SARS-CoV-2 to influenza and lots of others, directly disrupt parts of the immune response. So in a lot of cases, they're avoiding, or they're making sure that the host can't use cytotoxic T cells, or they're making sure that the host can't use the interferons. And then a lot of pathogens somehow get around phagocytosis. Either they survive phagocytosis, or they just prevent it completely. So those are ways hosts can avoid immune systems. And then these are the toxins. They're exotoxins and endotoxins. This is an important distinction to make. An exotoxin is excreted by bacteria when they're alive. Lots of diseases happen when bacteria excrete toxins while they cause an infection. Endotoxin is lipopolysaccharide, so it's a part of gram-negative outer membranes. And so endotoxin is released when gram-negative bacteria lyse or are killed, whereas exotoxins are released by live bacteria. So that is that's virulence, and then we can look at the host resistance factors. There are a lot of, this is like all of innate immunity, all of adaptive immunity, but also the health condition of the host. So stress, for example, can lead to cortisone release, and cortisone can suppress parts of the inflammatory response. This is one known way stress can lead to more susceptibility to infections, but there are lots of other things we see where a person who has had one disease is more susceptible to the next one. Yeah, things like diet, antibiotic treatment, anything meant to tamp down inflammation, these can all make a person more susceptible. When I was an undergraduate, our teachers told us a story of a person who had taken, I don't remember what it was, but it was some kind of over-the-counter treatment that decreases an inflammatory response to a bug bite or something like that. But this is a famous case, and this is not a rare thing, but a person was unlucky because the bug bite was infected also with a bacterium. And if that inflammatory response doesn't happen because of that anti-inflammatory medication, that bacterium can escape the innate immune response. And that's one way a healthy 20-year-old adult can be killed by bacteria. So think twice about using anti-inflammatory medications. Whoops. And I mentioned all of that. Okay. Then this is the second to last. Okay, and that's enough for this video. I'll see you with the next one.